Hi, my name is Hannah Schmidt, and I'm going to be looking at case 2 and case 4 for my case connections today. So to begin with case 2, in this case, a brain MRI shows a large glioblastoma tumor with necrotic areas. Adjacent to this MRI is a PET scan, which is shown after administration of fluorescently labeled D-glucose. So in order to determine how glucose uptake differs in the tumor as opposed to surrounding tissues, we will first look at normal uptake of glucose in different tissues in the body. So to begin, in the liver is permeable to glucose, whereas other tissues are not. Liver glycogenolysis is activated by glucagon and epinephrine. The liver supports blood glucose in the fasted state. GLUT2 is the mechanism of uptake, and GLUT2 is always present on the cell membrane. Insulin will inhibit cyclic AMP and activate glycogen synthase. Glucagon and epinephrine activate cyclic AMP, which activates glycogen phosphorylase and leads to glycolysis. In the skeletal muscle glycogenolysis is used. This is activated by epinephrine, AMP, and calcium. Glucose is oxidized as fuel by exercising muscle, which lacks glucose 6-phosphate and is not impacted by glucagon. Skeletal muscle uses the GLUT4 mechanism of uptake. And in skeletal muscle, glucose is not released from the tissue and does not contribute to blood glucose level. In adipose tissue, these tissues will store excess glucose as fatty acids. The mechanism of glucose uptake is through the GLUT4 insulin-dependent mechanism and uses stored triacylglycerols for glucose uptake in the fasted state. Now in the brain, this is a slightly different mechanism of uptake using the GLUT1 and 3 mechanism, which is insulin independent. The pathway for providing fuel in the fasted state is hepatic gluconeogenesis and glycogenolysis. However, due to the restrictive permeability of the blood-brain barrier and relative lack of local brain carbohydrate stores, the central nervous system relies heavily upon the blood-brain barrier expression of transporters for the delivery of key nutrients and solutes to the brain. Recently, it has been shown that lactate produced by astrocytes is a key energy substrate needed by neuronal cells. In order for astrocytes to produce lactate, glucose has to be transported across the blood-brain barrier and the astrocytic membrane via transporters. Glucose enters the brain extracellular space from the blood via the GLUT1 transporter present on membranes of the blood-brain barrier endothelial cells. Now to look here how the glucose intake in normal tissues differs from the glucose uptake by tumor cells in this image. So PET scans are used due to the augmented glycolytic activity of tumors for the identification of metastatic lesions. This technique takes advantage of the increased ability of tumor cells to take up and metabolize glucose compared to normal tissues. So as you can see here, the area of the tumor cell shows much higher uptake of glucose, and this is due to the increased glucose consumption due to the overexpression of GLUT transporters and hexokinase. This is known as the Warburg effect. This effect says that cancer cells prefer aerobic glycolysis over oxidative phosphorylation. However, further research has shown that cancer cells engage in both aerobic glycolysis as well as oxidative phosphorylation. So now to see why this would be advantageous to an invasive tumor. So like I said, the tumor uses both aerobic glycolysis as well as oxidative phosphorylation in order to generate energy stores, and the cancer cells maintain high rates of both this oxidative phosphorylation and glycolysis. And this is to fulfill high demand for anabolic processes and to feed branching pathways that generate nucleotides, amino acids, and fatty acids in order for the synthesis of nucleic acids, proteins, and membranes. Cancer cells often exploit these regulatory pathways to fulfill anabolic needs and to adapt to various microenvironments. So modifications that may have occurred in cancer cells to alter the glucose uptake. To begin, one function of oncogenic pathways is to drive cell autonomous nutrient uptake and program proliferative metabolism, whereas a function of tumor suppressive pathways is to prevent nutrient utilization for anabolic processes. In this instance, so for cancer to arise, mutations are needed to give the cells the ability to acquire nutrients and coordinate regulatory metabolic pathways to support their proliferation. And for example, there is a large percentage of human cancer cells that are dependent on aberrant signaling through the PI3K-AKT pathway. And there's a lot of evidence showing that this pathway causes increased dependency on glycolysis. And in this particular example of the glioblastoma, there's 12% of these tumors that harbor the same point mutation in the gene encoding cytosolic isocitrate dehydrogenase 1. Isocitrate dehydrogenase 1 coupled the interconversion of cytosolic isocitrate and alpha-ketoglutarate and an NADP plus NADPH dependent reaction. So there's not exactly clear effect that a lot of these mutations in cancer cells have currently. However, it's known that there's oncoproteins that are known to either elevate expression or induce activity of enzymes and transporters that facilitate high rate of glucose metabolism in cancer cells. Now to move on to case 4. Case 4 is a 2-month-old previously healthy female presents with jittery spells after breastfeeding. The baby hasn't had anything to eat other than breast milk since birth. And these symptoms began when the baby began sleeping more than 2 hours between feedings. Lab results indicate hypoglycemia, and this shows that glycolysis is inhibited or impacted. Epinephrine is administered with no increase in blood glucose, but an increase in blood lactate. So the liver is also 4 centimeters below the right costal margin, indicating hepatomegaly. And the blood glucose will increase after breastfeeding, but the blood glucose can't be maintained in normal range during fasting, nor indicating the fasting state glucose metabolism is impaired. So in order to determine which is the most likely diagnosis for this infant, we'll look at the different options more in depth. Option A is hereditary fructose intolerance. Hereditary fructose intolerance is a condition that affects the ability to digest fructose. This disorder involves a deficiency in aldolase B, which is affected by the ALDOB gene, and mutations in this gene reduce the function of the enzyme, resulting in the accumulation of fructose 1-phosphate. Fructose 1-phosphate is then trapped in the liver, which results in hepatomegaly and fasting hypoglycemia. This inhibits glycogenolysis and gluconeogenesis, and aldolase B is the rate-limiting step for fructose metabolism. Those affected by this condition develop signs and symptoms of the disorder in infancy when fruits, juices, or other foods containing fructose are administered. After ingesting fructose, they will experience nausea, bloating, abdominal pain, diarrhea, hypoglycemia, as well as failure to thrive. However, this infant has ingested exclusively breast milk. Breast milk is composed of lactose, which is composed of glucose and galactose. This disorder is unlikely because the infant has not consumed any fructose. So we can eliminate that option. Option B is galactosemia. Galactosemia is a disorder that affects how the body processes galactose. Galactose is taken up by the liver and phosphorylated to galactose 1-phosphate. Afterwards, it is activated to UDP glucose. The enzyme responsible for this is galactose 1-phosphate uridyl transferase, known as GALT. In normal metabolism, UDP glucose can then be converted to glycogen or to glucose 1-phosphate, which can be converted back to glucose 6-phosphate, where it can enter the glycolytic pathway. In galactosemia, galactose 1-phosphate is elevated in the liver. This inhibits hepatic glycogen metabolism, as well as other pathways requiring UDP glucose. Accumulation is toxic to the liver, kidneys, and central nervous system. Infants with galactosemia can have life-threatening symptoms that usually appear a few days after birth. This infant has been more than 2 months, and the symptoms began only when the baby began sleeping more than 2 hours between feedings. And so this is unlikely in this case. We'll move on to look at the third option, which is glycogen storage disease, known also as von Gurk disease. This is an inherited disorder caused by the buildup of glycogen in the cells. Mutations in several genes prevent the effective breakdown of glucose 6-phosphate. Glucose 6-phosphate that is not broken down to glucose is converted to glycogen and fat, so it can be stored within cells. Accumulation of glycogen can be toxic to a cell and damages organs and tissues throughout the body, particularly liver and kidneys. Glucose 6-phosphatase is the enzyme required to dephosphorylate glucose 6-phosphate to generate free glucose. This is used both in gluconeogenesis as well as glycogenolysis. Epinephrine will increase cyclic AMP, which phosphorylates and activates glycogen phosphorylase. Glycogen phosphorylase activation results in degradation of glycogen, glycolysis. This results in free glucose being released by glucose 6-phosphate, being dephosphorylated to glucose. This is under normal conditions. In this case, if epinephrine was administered, free glucose would not be able to be released due to the inability of glucose 6-phosphate to be converted to glucose via glucose 6-phosphatase. Glucose 6-phosphate then accumulates, which results in glycogen accumulating. So if epinephrine was administered as it was in this case, glucose 6-phosphate won't be able to be converted to free glucose, but glucose 6-phosphate can go on to glycolysis and pentose pathway, which is why lactate would be produced, but not glucose. So signs and symptoms of this condition typically appear around age 3 or 4 months when babies start to sleep through the night and do not eat as frequently as newborns, which would be the case with this infant. The affected infants will have hypoglycemia, as does this infant. Lactic acidosis can appear, as well as high uric acid levels in the blood and hyperlipidemia, as well as hepatomegaly, which is seen in this disorder. This is a likely option for this infant, as I said, due to the epinephrine administration, in this case, hepatomegaly and fasting hypoglycemia. However, we'll look at the last option, which is glucose 6-phosphate dehydrogenase deficiency. Glucose 6-phosphate dehydrogenase is used in the pentose phosphate pathway. Glucose 6-phosphate dehydrogenase is required to convert glucose 6-phosphate to ribulose 5-phosphate. NADPH is produced in each reaction. The pentose phosphate pathway is the only source of NADPH in red blood cells. The loss of glucose 6-phosphate dehydrogenase can lead to hemolysis and jaundice due to a decrease in NADPH production. This would present as hemolytic anemia, which occurs when red blood cells are destroyed faster than the body can replace them. This would result in paleness, jaundice, dark urine, fatigue, shortness of breath, rapid heart rate. Reactive oxygen species would also accumulate and damage red blood cells in this case. This condition is an inherited X-linked recessive pattern, so males are much more likely to experience this disorder, and it's seen usually exclusively in males. In this case, this is a female infant, so it's very unlikely in this case. And the infant also doesn't have any signs or symptoms consistent with this disorder and does not have a condition where the pentose phosphate pathway is impacted. So we can eliminate this disease as well. So the answer will be C, which is glycogen storage disease, also known as von Gert disease. This will conclude the case connections.